rules to infer a relationship between pseudogene segmental duplications. There were a couple of goals that we had with this project. First of all, we wanted as possible to use the formal reading tools provided by the Semantic webs to provide biologists the ability to categorize information and to draw meaningful inference from it. And also to show that the study of the relationship between segmental duplications is the application of this technology. Paper that I'm going to refer to is the conference paper, listening on the authors. And I'm also going to draw from a little from my colleague Rana's paper, Segmental Duplication in the Gene for Details of Pseudogenation. So I think I'll to talk about biology and by definitions and definitions for what we're accomplish with the reasoning. Pseudogenes in the genome that are derived from the gene have somehow become stable over the evolutionary history. This disabling occurs as a result of different copying events. We usually categorize pseudogenes into two main types, duplicate pseudogenes, which arise from block. In this case, there are mutations which result in a lack of loss of function in the gene. The second major type is process genes. These are transitions. They lack promoter sequences and usually are fentrons in the presence of a 3' poly A tail. Duplications are relatively new duplication. Usually they occur in the last 40 million years or so. Usually we define them as having a nucleotide sequence greater than 90% and are long in length. And these regions are notable for higher structural, including large amounts of insertion and deletion. So motivation for studying the relationship between genes. First of all, notable that there's a high enrichment of other genes in regions. For example, about of consistency in this occurs 70% of genes, 80% of all pseudogenes. In general terms, study of this interrelationship provides us with insights and genomic evolution and to better gene annotation, information about how and when other genes were formed. Specifically, for example, we discovered cases not from actually from the duplication of their pseudo. The second course we kind of looked at is to substitution rate and the surrounding intestine, and this allowed us to about when disablement of pseudogene occurred with respect to when. To have this, found it useful to create kind of a formal tree-like structure consisting of hierarchy of pseudogene. As you can see from that diagram, we created the hierarchy by entering and classifying pseudogenes in this manner. Beginning, we looked at all of the pseudogenes, and these are human pseudogenes, and gives counts of the number of pseudogenes which fall into these various categories. But first, we had pseudogenes that occur in SD roots. Then we asked if the gene of the pseudogene occurs in an SD read. Then occur in two segments E pair, and then we look for if in addition to parent genes, also cis pseudogenes, and then if there are, then we look at determine if our pseudogene in question arose as a result of the duplication of the parent gene or of a sister gene. And if not, we can look at the comparison of the nucleotide spin rate transitions about when the pseudogene arose and became old. Looking at a little more example, we're looking at pseudogenes which were as a result of duplication of pseudogenes. And this is because there are often cases in SD pairs there are more than one pseudogene which are parent genes. We call these pseudogenes. And this is a nucleus of pseudogenes. First, we called duplicated process. In this case, we have pseudogene rose from a retrotransposition event, and then were duplicated. So they're process genes which are duplicated or duplicated. Have duplicated, which were duplicated pseudogenes, which were then doing as part of the second duplication. These types of distinction are not found in pseudogene called algorithms. They're not duplicated pseudogenes, and especially notable for the process because it can lead to difference in how they're annotated because the pseudogene was a result of a block duplicate. Initially, it arose in exposition rather than a block duplication. We're looking at pseudogenes and parent genes occur in the Semantic pair, and we pair the nucleotide substance, the pseudogene in comparison with its parent region. And the comparison we use is the distance score. Which is distribute, and we found that 4 could be significantly high, and values less significantly low. We found this is the substitution rate was similar, and the duplication events immediately the decision, and we see neutral substitution. We did find a couple of types of discrepant cases where the pseudogene subs were significantly than the surrounding SD region. Higher, it could mean current things. It could mean there's positive select or negative select large region. Substitution rate indicates the stability of negative selection of the pseudogene. Perhaps most interestingly, that perhaps a pseudogene functional. Okay, so this kind of suggests a formal reason useful. And, what we decided to do is create knowledge and a series of prediction rules to do a classification scheme, and also the resulting inferences that could be made using it. So the technologies are kind of the standard semantic web. So just want to have a brief through of this web stack. The flow is the basing is the uniform identifier. These provide unique IDs for any fine enough space, which are based upon resolvable address on the web. RDF, the resource description framework, provides a language, describing terms. It's formed triples in a credit object, and it's typically serialized as XML or InchIb. RDFS provides provides us the ability to find classes and keys for these classes. The web ontology language or OWL us more richer analytical terms to create more rigorous. Then there are query languages. The most prevalent is allow us to questions from our knowledge base. That it developed as a relational knowledge base. We have types of languages, of which the most common these days is semantic web. This allows us to define rules, which are written rules, and can use to further instances our knowledge base. And I think it's a unified object, which in the semantic web rule is a description line. And this allows us to perform formal reasoning and prove the definitions. One of the things of the semantic web is that it allows ontology to be used and extend. And it's important when working with the technology, work with ontology, and result. A lot of has been done in the biomedical community, reuse ontologies to various medical. Of these particularly notable is the sequence ontology, which is and relationships which sequence ontology is basically three major hierarchies. First of all, feature. These are elements on the gene, and can range from anywhere as small into larger branches of genes. Sequence variables are class variables, such as alleles or copy number variation. We have semantics, which are used to describe aspects of either sequence features or sequence variants. These are sort of things like conserved, retroposed, and these are used to label elements on it. We decided for to create a use ontology and add terms where necessary. So this gives indication terms we borrowed and extended the existing term. And these properties also were added. As you can see, SOR contains property pseudogene. Added further subclass for duplicated pseudogene. We created a property parent gene, which is a property of SOR, which is called functional homolog of, and we find pseudogenes to be which are protein coding genes. Although it is different protein coding genes, with pseudogenes through, so in all cases they are genes going to be protein gene. SCBs are two of these segments, and this is created as a subclass or low-level SO term called biological region. And then there's a reciprocal S pair of which extends the SO term similar to. That's to create S pairs. Containment of genes or pseudogene within an SO region in these properties contains pseudogene and contains gene. These are extensions of the SO part. Another extension was in terms of attributes. Our sequence attributes describe our descriptive properties, and they indicate things which are either generally present or absent, or essentially Boolean properties. And what we want to do is also to describe numerical properties, such as the Kimura score or count. So what we did was to create a subclass of attribute, and this is a numerical view. Subclass to refer to specifically properties in gene class numerical attribute. Third area of extension is to handle specific location sequence on the genome. This discussed in it's really important to be able to decouple sequence features from their location on the reference build. And as we know, the build often changes, so it's also possible to describe individual organisms. Of course, the exact location. So, generally, to external data, it's typically this is typically handled in the database bowl. Then the Chado architecture has a means of doing this. For our building, a knowledge base in now, we actually need to create a class to hold this information. So we create a class nucleus and we use the property located and for these where an instance of in which they're located. And the new class has properties which can describe the build, the chromosome, and information like that. Reflect a knowledge base, and we can populate it with individual genes and these. Now, I wanted to create a set of rules which could be used to classify the pseudogenes from them. So this diagram shows kind of a flow of how these rules should be applied, and it's the same structure diagram which I showed earlier. Except it does indicate where the rules take effect in the examples. Take a look at an example, kind of arbitrarily rule 4. In this case, we're looking for pseudogenes which may be under negative solution. And this reflects the whole rule. It's actually written in kind of a sparse pseudocode. The actual swirl is kind of a XML serialization, which we actually did program the old API. This rule does essentially is it looks for individuals. It finds the parent gene, it finds the segment that the pseudogene finds the duplicate of the SD pair, and given that the parent gene is located in that duplicate segment, make sure that there's not a pseudogene in the same segment and the gene. Looks at the Kimura scar to compare the aids with its surrounding region. And if the value found to be significantly low, then it's considered as being under negative selection. We can compare that and what the paths followed on the decision. Secondly, a more complicated rule is, in this case, we're looking for pseudogenes of a sister pseudogene rather than parent gene. This makes use a lot of the swirl language, which allows you to do arithmetic. For both, but what we're doing here is we're looking at pseudogene in which both parent this pseudogen located on duplicate of the SD pair. Look at the distance of compare the distance from the star of the SD pair and the sister pseudogene to determine which it aligns. And if a better sister pseudogene, then we determine that it was more likely to the that, and we give it aligns with another pseudogene. This figure kind of illustrates first we have the path of the decision tree, and then in this figure is kind of a look overview of the sequence. Like this is the pseudogene in question. This is the duplicate of the SD pair, and this is the parent gene, and this is the pseudogene. And as you can see, the alignment is much closer with the sister pseudogene than it is with the parent. So in this set of rules and given a fully populated knowledge base, we can have a full set of analyses using OWL reasoner, and we have a full set of analyses we code into a store and run query. We found the process of generating entailments to be very simple in terms of memory. It took about 30 minutes, but it consumed around 32 gigabytes of RAM. So, the advantage, once we have a full set of entailments, query to run and very fast. And see from these examples, they're very, very simple queries. Here, this shows the ontology that we're working with fit in with existing sequence ontology and also possible future directions. What we have now is kind of a useful prototype that we can perform against, but it kind of stops short of being a full ontology to describe pseudogenes. And we think it might be useful to create such an ontology in the future. And possible terms we might want to add. We might different characteristics of pseudogenes, whether they're transcribed or regulatory. Also, they're possibly the origin of the pseudogene, whether it's from the nucleus or mitochondria. And we can also look at features of segmental duplicate characteristics, whether being long. So I would like to thank my co-authors, especially Dr. Karan, really trying to know about pseudogenes and SDs. I thank the Yale program, which is really amazing program, and also the ISCB, the Department of Energy, and the National Science Foundation for making this possible, providing partial support in the form of a travel grant. And I'll take any questions.
About the open and plural decisions.
Certainly.
So you're assuming an open world decision, but I assume that if a pseudogene is in one class, it can't in another class, or do you do that explicitly, or how does it work?
Explicitly.
Yeah. So you showed how you're calling the different types of pseudogenes.
Yes.
But if you don't know it's not one of the ones, you know, in a closed world, you're doing that it's not one of the other ones if you know it's one, if it's type A. But in the open world, you just don't know.
Well, we do
but
make
you
distinction.
It's in rules to say that if it's type A, it can be type B or C.
Right. You're referring to the distinction between duplicated and processed pseudogenes?
Well, it's all your different distinctions. So in open world assumption, you don't get that for free. You have to put it in. Because you just don't know unless you tell it. Whereas in a closed world, you would, type A, it wouldn't be of type B.
Yeah.
How does that work?
Are you referring to the deductions that are reached from the rules?
Yeah.
I'm not quite following.
Okay, well, if you infer that's of type, can it also be of type B?
Yes. Oh.
So they're not mutually exclusive. Okay. Discuss it later.
Okay.
A simple question, one which goes a similar direction, but it's not here. So I think the key question is, do you load all genetic information in your triple store, or do you use some kind of other stupid data resource for genetic information? So, for example, where a gene starts and ends, and
Oh, we do keep that in the triple store.
Yeah.
Yeah, it's used by the one of the rules.
Where do you pull it out?
It's from the reference build.
Mm-hmm. Then the other thing, and once you showed that a pseudogene is part of a protein encoding, right? And then you show that a pseudogene can be duplicated, but it doesn't mean necessarily the duplicate gene is in a protein encoding region. Is this the case? Or? This might be.
I believe that's the case. Yes.
Okay.
Thanks. Okay. Thank